Alright, everybody. Hello. Welcome to the final Biochem in the 3-course series of the Biochem 44X group. This is Biochem 442. It's the last one of the series, and, oh gosh. Been on a long journey with you guys. So this is the final first lecture that you will see from me. At least in this series. Who knows if I'll decide to do things for PChem and whatnot. But yeah, let's go ahead and get into this. And yeah, okay, so 442, I'm gonna skip all of this, okay? Professor Kong, I've heard is very sweet, very nice. I had a couple interactions with her over email specifically. She seems amazing. The last prof I had was Dr. Andrea Willis. She was also fantastic. I also had Professor Rohola Baker, also fantastic. The slides are a bit interesting for Professor Rohola Baker, but she's very enthusiastic about what she's teaching, and I thoroughly enjoyed her section. Despite 442 probably being my worst grade of the 3 biochems, I found that it was a pretty cool class. So go ahead and I don't really care about all this stuff. I really want to get down to the content, and the content starts around here. Yeah, so let's talk about a couple things. So what makes a liver cell different from a neuron? There's a couple things. What mRNAs are expressed, what proteins are expressed. Things like expression of enzymes, like where are they expressed, are they even expressed at all? There's all sorts of structural components like microtubules, like neurons have a lot of microtubules, maybe a lot more than some other types of cells, like maybe hepatocytes don't have as much microtubules as neurons, things like that. There's also, different environments, liver cells are quite close to each other, stuff like that. There's actually like what cells do they receive signals from, all that stuff. You guys know this stuff pretty well, I hope. The composition of a cell is made of the bilayer, hydrophilic outside, hydrophobic inside, and then the inside we've got a bunch of things. We've got the nucleus, cytosol, and then we got the organelles. Specifically, the organelles for eukaryotic organism. Prokaryotes don't have organelles. But you guys are going to focus a lot on the ER in these first couple weeks, and then eventually you actually start going to the ER and the Golgi specifically, and then you'll eventually switch to the mitochondria when you do apoptosis and stuff like that. But you guys know all this stuff pretty well at this point, I think. This is just a slide to show that the cell is very crowded. It's a very compact and small machine. It's very efficient what it does. So in 442, you're going to talk about, these things on this list. I'm not going to go over a single one of them, but specifically in the first 5 weeks, we're going to look at a couple of things. So we're going to look at mRNA molecules, their lifetimes, how we determine stability. We're going to look at microRNAs, initiation factors, ribosomal sequences, stuff like that. So the first thing that we look at here is obviously, central dogma and life of protein. So there's a couple things that can happen. Once we've transcribed, we make mRNA, and then we translate, we make a protein, and then from there we have a protein that folds, and then we can post-translationally modify it. So we can phosphorylate it, we can do all sorts of weird things to it, and then after we do those modifications, we can do a couple things. We can either maybe some of them help with the cytoskeleton, maybe some of them help with cell cycle, maybe some of them are secreted and use as signaling molecules for other cells, maybe some of them are enzymes, a whole bunch of things. So the first thing we're talking about is, translational control. And so there's a bunch of ways that translation is actually regulated and controlled. So, there's a lot of different translation factors. So there's proteins, there's, amino acids. So, proteins can regulate translation. A lack of amino acids can regulate translation. A lack of tRNAs can regulate translation. And specifically here, initiation factors. If you remember from 441, there's IF2, IF1, IF3. Those initiation factors are helpful. There's also, the access of translation factors to mRNA. So, can these factors interact with the mRNA, and then there's also, you know, how stable is the mRNA in general. So like we're looking at three different things here, or a couple different things here. Do we have the necessary machinery to do translation? Do the mRNAs have access to that translation machinery? And are the mRNAs stable? So in particular, specifically, we're going to look at, you know, do mRNAs have access to a ribosome? And then stability-wise, we're going to look at, do mRNAs have a cap and poly A tail plus the circular structure that keeps it stable? So here is the example of that circular structure that stabilizes the mRNA. It uses a bunch of polyadenylate binding proteins plus some other initiation factors. We've got 4E and 4G here, along with the 5' cap that helps stabilize the whole thing. So, general translation or regulation of translation is, you know, regulated by initiation factors at the eukaryotic level. So, here you've got your EIF2, and we're going to be talking about this a lot, but essentially what happens is basically what's shown in this image. So this EIF2 basically binds to this massive complex that has the mRNA. And, you know, once that's bound, this EIF2 basically scans along the mRNA until it finds this AUG. And then once that's there, we use an ATP. I think it's actually described as a GTP in 441. But basically, this is used to basically attach the AUG, the tRNA to the AUG, if I'm not mistaken. And then from there, we actually use this. We use this GTP on the EIF2 to act as a signal to bring down the large ribosomal subunit onto the small ribosomal subunit. So, you know, in this step here, we're just attaching the ribosome, the small ribosomal subunit, to the mRNA. In this step, what we're doing is we're hydrolyzing an ATP to kind of cause the movement. So the movement is caused by ATP hydrolyzation. And then in this step here, the hydrolyzation of GTP causes the methionine to basically stick to the start codon, and then it also recruits the large ribosomal subunit. And then from there, we can continue the process of translation. So, one of the big things to keep in mind is that if I get rid of this EIF2 GTP, I can't do translation. So there's a couple of things to keep in mind with this. So how does EIF2 get back the GTP after it's been hydrolyzed? And the way that it does this is through a guanine nucleotide exchange factor, otherwise called a GEF. And this is the GEF for EIF2 is EIF2B. Pretty easy to remember. This is a thing that takes a GDP and replaces it with a GTP on the EIF2. And so here's a nice diagram of that. You can see here the GDP form. It's going to bind through some interactions like hydrogen bonding and stuff like that to the GDP version of EIF2. It's then going to kick off this GDP. It doesn't add a phosphate to the GDP. It kicks it off. It kicks off the GDP, causes this thing to result, and then it adds a GTP. And so after the GTP is added, it actually causes a dissociation. So like this GTP is added, it causes these two things to split. And so then you have a new EIF2B that can be used to recharge another EIF2. So under conditions of stress, one thing to keep in mind is translation doesn't proceed without EIF2 GTP, which makes sense because you need to start the whole process. And so under normal conditions, you have this process. But under stress conditions, what you actually end up doing is you actually end up phosphorylating the EIF2 GDP. And what that causes is the EIF2B to bind so strongly to the EIF2 GDP with the phosphate that they just don't dissociate. And so basically what ends up happening is that over time, all of these EIF2Bs are sequestered, and you can't regenerate any EIF2 GTP, and so you actually stop translation. This can be reversed by just removing the phosphate group, but when there is a phosphate, it stops translation completely. So this is talking specifically about translational control at the ribosome level, but let's talk about translational control at the mRNA level with mRNA stability. So, general reduction of mRNA stability is dictated by the poly A tail. So this should be pretty self-explanatory. We've got a 5' cap, we've got a poly A sequence, we've also got this complex here. These help stabilize the mRNA so that when you want to translate it, it doesn't just explode. So there's a couple things that could happen. So you can see here, over time, generally what happens is this poly A tail degrades. This should hopefully make sense because remember, RNAs have an extra 2' hydroxyl group, and so it kind of self-hydrolyzes itself over time, which is why this poly A tail exists in the first place, actually. It mostly exists there so it can, self-hydrolyze itself, and it keeps the rest of the mRNA intact. Over time, you're going to get poly A shortening, so we go from 200 to 25. And then from here, we actually have two options. We can either decap the mRNA, so remove the 5' cap, and then follow that with a really quick 5' and 3' degradation, or we can just continue to degrade from the 3' end. So, honestly, you guys don't need to know when it degrades from the 5' end and when it degrades from the 3' end. You just have to know that these are two different options. It can either remove the cap and degrade very quickly from this side, or it can degrade normally from some standard rate from the poly A tail. There is also a couple other things to keep in mind. So a couple things can happen, right? So if you want to translate something, you obviously would make this complex, but it's equally possible that these are in equilibrium with each other. So you can have this form, or you could have the form where a deadenylase actually attaches on to the 5' cap and actually starts chewing the poly A tail, resulting in degradation. So it kind of depends, right? So, maybe if you have a lot of complexes, you do this, but if you have less complexes, maybe you make this. So it kind of depends. There are also things, within certain mRNAs. So mRNAs can make secondary structures. They can make hairpins and stuff like that. And these can change their translation stability. So some mRNAs actually make secondary structures, and those secondary structures bind proteins, and some of those proteins can either prevent translation or promote transcription, so it kind of depends. And an example of that is actually here. So if you look at this, this is something I'd actually remember for your exam. It actually could show up. So regulation of mRNA stability example, so we got transferrin, which is a fun little enzyme. And so if we look at this, we can see here that iron levels are pretty precisely regulated by the body. And so we are looking at under conditions of iron starvation and excess iron. We've got ferritin and transferrin. So ferritin uses iron and transferrin imports iron. So if you have a lack of iron, you're going to use transferrin, and if you have too much iron, you're going to use ferritin. And so we also have aconitase. So aconitase, remember, is actually like the first enzyme of citric acid cycle. It's the same enzyme. It's a dual function protein that promotes iron import. It prevents ferritin translation and stabilizes transferrin mRNA. So what we're going to do here is we're going to take a look at this. So under starved iron conditions, you're going to want to have the transferrin protein made. So what's going to happen is under starved conditions, the aconitase binds to the 5' hairpin, and it stops translation of this ferritin, because you don't want to use iron, you want to absorb more iron, so it actually blocks it here. On the other hand, for the transferrin mRNA, there is a 3' hairpin, and this 3' hairpin actually acts as a shield to stop degradation. So when this is there, it prevents degradation, and therefore translation of this mRNA can occur more readily. When there's excess iron, though, the exact opposite happens. When there's excess iron, this 5' hairpin is no longer blocked by a aconitase, because aconitase binds the iron in this spot. And so that allows for translation of the ferritin, so you can use the iron. But in the case of the transferrin, the binding of the iron to the aconitase opens up this cleavage site so that you can actually cleave this mRNA and break the mRNA so that it can no longer be translated. So it depends on the situation. And there are many different examples of this, but this is one of them. Another example of translational regulation is microRNAs. And so microRNAs are a bit interesting, but here we have an example of I think how a microRNA is actually made. And so what you've got here is eventually, you're going to translate something from your genome, and then what happens is, to make an mRNA or to make a microRNA, what ends up happening is you take this RNA that you made, you chop it up, and then you keep chopping it up. So you crop it and then you dice it. So you crop it in the nucleus, you dice it in the cytosol, and then you basically take this protein. What the heck happened to my? You take this protein called an argonaute, and other proteins as well, and you attach part of this large RNA portion onto the argonaute protein. And this is called a risk factor. So this is or a risk complex. I can't remember what the risk stands for, actually. I think it's RNA-induced silencing complex, I think, something like that. It's not too important, but basically what happens is this protein binds a piece of RNA. And so two things can happen, right? Once this has happened, this is now a complex that silences RNA. And so there's two options, like I said. You can either have a very extensive match where this RNA portion pretty much directly matches a section of RNA on an mRNA molecule, or you can have it match a little bit, not too much. So let's start with the extensive match. If it matches a lot, what ends up happening is that basically immediately leads to degradation. So it matches, causes slicing by the argonaute protein. The argonaute protein basically cuts the DNA, or sorry, not DNA, cuts the RNA, and then using a piece of using ATP, ATP allows the hydrolysis of ATP allows for the breakage of the RNA, so into nucleotides, and then the RISC is released for reuse. So now you've got a cleaved mRNA and a reusable RISC. If you have a less extensive match, what ends up happening is this complex binds, but it doesn't degrade super quickly. It binds and then blocks translation, and by blocking translation, it eventually leads to degradation over time. So this is a much slower process. So like you said here, rapid translational repression, because it's physically blocking it, and eventual degradation. It's not like this, where it's rapid mRNA degradation, right? So complete match, rapid degradation, slight match, repression into gradual degradation. That's how to remember that. So, here's a couple things to keep in mind about microRNAs. MicroRNAs, they can have multiple targets. So one microRNA can bind to many different mRNAs. You can also have multiple microRNAs bind to one mRNA. No one says that you can't bind multiple microRNAs to one mRNA. And also, microRNAs can be cell type specific. So maybe there's a microRNA in a muscle cell that's not in a liver cell. So example, maybe you have what was it called? Glucose 6 phosphatase, for example, right? That is an enzyme that's only in liver cells. Maybe you, for muscle cells, maybe you have a microRNA that prevents translation of glucose 6 phosphatase mRNAs, and so it stops translation of it. But in liver cells, you don't have that microRNA because you need glucose 6 phosphatase. So, no microRNA, make the enzyme. But for muscle cells, have the microRNA because you don't want to make that enzyme. So that's actually the end of this lecture video. It's the first one. I still have to go through 5 more. So I will record those as soon as possible. But I hope this video was helpful to you guys. And yeah, happy studying and good luck.